Hello, I am Muttaqi Ahmadaladin and I, along with my colleague Bangaru Arvind, have worked on the project Plantpathotar. This project was done in partial fulfillment of the requirements for the degree B.Tech Biotechnology at the Chaitanya Bharati Institute of Technology under the guidance of Dr. P.V.P.S. Arun. Plantpathotar is an integrated repository of new drugs and drug target information of plant bacterial pathogens. Before we dive in, we can all agree that food is extremely important for our survival and that there are many people who do not have adequate access to food. Despite this, oftentimes we see headlines like this. On one hand, we do not have enough food, and on the other, we are losing food to diseases. These diseases, in part, are responsible for food shortages. A huge reason for this problem is antimicrobial resistance, due to which existing antimicrobial agents are not able to control pathogens. If nothing is done about this, this problem is only going to get worse as drug-resistant pathogens become more and more prevalent and as our ever-growing population continues to increase demand for food. In this project, we decided to address this very problem by finding alternate drugs that may be used to target these antimicrobial resistant pathogens. So, we can broadly classify our objectives in this project into three categories. The first objective is to predict drugs. These drugs are extremely pathogen specific. That is to say that these drugs target a pathogen without causing any adverse effects to host plant or humans eating that particular plant. The second objective is to develop an open access, user-friendly, easy to use online database wherein this information about the potential alternate drugs and drug targets can be stored. Moreover, we have also provided a portal wherein the users may submit additional pathogen host pairs for us to carry out the analysis and publish on our database. The entire website has been designed keeping ease of use in mind, as we realize that there may be many biologists who might not be as tech savvy. Coming to the importance of this project is extremely important as it is a sustainable way to manage diseases. Furthermore, since this is an in silico method, it's extremely quick, and its speed is extremely important in cases of pandemic, wherein new drugs need to be found and need to be found quick. Moreover, the drugs that are contained in this database are FDA approved, so repurposing them is relatively easy. Moreover, this particular project will aid scientists in wet lab studies. Now, let's talk about the way in which this process works. So, this DNA, which through the process of transcription gives rise to RNA, which by the process of translation gives rise to protein. Now, these proteins carry out various functions in the body. Now, in this entire process, if any stage is stopped, the pathogen is affected negatively. In this project, we are targeting proteins. We are targeting proteins and inactivating them so that the bacteria dies. There are protein targets and there are drugs. If the drug is specifically designed for a particular target and specifically binds to that target, it produces an effect. In case of antimicrobial agents, this effect usually is inactivation of the target protein. There are a huge number of proteins in the proteome of a pathogen, and using in silico subtractive hybridization, we are going to try to filter these. So, in silico subtractive hybridization basically refers to the process of identifying the common proteins between two organisms. Once these proteins are identified, they may be classified as proteins unique to organism A, proteins common between both the organisms, and proteins unique to organism B. Using in silico subtractive hybridization, we have developed a methodology which selects a subset of the pathogen's proteome. The drugs which bind to this subset are such that they do not have any effect on the host plant or human life but decrease the infectivity and the survivability of the pathogen. Drug bank database contains a list of drugs and their respective targets which have been experimentally verified. If any of the proteins in the pathogen are extremely similar to the targets in the drug bank database, we can hypothesize that the same drug will act on this protein too. Such proteins and drugs are stored in the database. Till now, we have identified drugs and drug targets in 8 agriculturally important pathogens, and on average, there were 4,226 proteins in the proteome of the pathogen, out of which 94 were found to be known drug targets, to which 123 drugs were found to bind. Demonstration of Plantpathotar. Here's the homepage of the Plantpathotar database. It contains a brief description of the database in the center. To the top, the header contains links to various pages in the database. To the left, there's a list of all pathogens currently in the database. Selecting any of these leads to its summary view. For example, if we select Agrobacterium vitis, we are taken to its summary page. Here, information such as the number of proteins in the pathogen proteome, the number of drugs found, and the number of drug targets found can be seen. Links have been provided to download this summary table as well as the proteomes of the pathogen and the host. Moreover, there are external links to get more information about the pathogen and the host. The list of drugs can be accessed using the Drugs Found option. Selecting this option leads us to the simple view of the Drugs Found page. Here, we can view the list of all the drugs found along with the respective PubChem IDs. External links have also been provided to facilitate the user to know more about the drugs. Clicking on the PubChem ID leads to the PubChem listing of that particular drug. Clicking on the name of the drug leads to the drug bank listing of that particular drug. Switching to the detailed view, we can see the name of the target and its information along with the drug details. All these tables can be downloaded as CSV. We can also search the database on drug query using the search option. For example, if we search using the drug ertapenem as query, all pathogens on which this drug acts are shown in the results along with other related information. Finally, let's talk about the Submit Proteomes option. To access the Submit Proteomes option, you need to log in first. On logging in, the Submit Proteome option appears in the header, selecting which leads to a Google form where the user can enter the information and upload the pathogen and the host proteomes. After the analysis is carried out, the results will be uploaded to the database. To conclude, the Plantpathotar database will aid researchers to select new drugs in case of resistance to existing drugs. Moreover, this process is extremely quick and will be extremely useful in cases of pandemic. Additionally, this particular database will be extremely useful to scientists in carrying out wet lab screening of drugs. Thank you and have a nice day.